Or is that for
Okay.
for Facebook or?
For the Facebook.
Yeah. Actually, one thing is not correct because my main affiliation is actually Naturalis Biodiversity Center.
Oh, okay. We will correct that later.
Yes. Okay. No problem.
Okay, okay, so I guess I think the participants are just coming in, so maybe we can start. Okay, so good afternoon, everyone. Welcome to the third Philippine Planktology lecture series. Just last week, we had a talk from Dr. Maxine Mo from National University of Singapore, and then today we have our second speaker. And so I hope we are all excited. I think the participants are just coming in. I will introduce our speaker in the next slides. Okay, so the Philippine Planktology lecture series has been running for 3 years already, and so far we have invited the best of the field. Okay, so I hope that we will learn from these lectures. Okay, so just a short introduction of our program. Starting at 2 o'clock, we will just show you the house rules, and then we will introduce the speaker, and then maybe a bit more time for Dr. Katja to give her talk, and then maybe 10 to 15 minutes of the open forum. So I will be the moderator instead, or the discussant for this talk. And then we will award the certificate to our speaker. And then if we still have time, we can have a photo taken. Okay, so sorry, I cannot see my controls here. All right, so I think everybody is familiar with the house rules on online webinars. So I would like to ask everyone to please turn off your cameras and then your microphones on mute. And then if you have to ask a question, you can chat in the question and answer box. And then you may hold your questions until the end of the presentation, and they will be entertained during the Q&A portion. And then if you have to ask your question live, it's okay. Just raise your hand and wait for the moderator to call you in. And then at the end of the talk, and then the awarding of the certificate, an evaluation link will be sent to the chat box. And please accomplish this so that you may receive your e-certificate. All right. So, okay. So today I would like to introduce to you our speaker. She has developed a unique research line at the interface of ocean science and evolutionary biology, applying cutting-edge morphological and molecular tools to study zooplankton. And she currently holds a group leader position at the Naturalis Biodiversity Center, which corrects the poster. Okay, so she is now affiliated with the Naturalis Biodiversity Center, which houses the Natural History Collection of the Netherlands, and she is an associate professor at the University of Amsterdam, where she teaches and coordinates the MS courses in biological oceanography. Please help me welcome Dr. Katja Peijnenburg.
Thank you, Christy. Wonderful
Yes.
introduction. So I will start sharing my screen now.
Yes, please.
This will stop others. Yes, okay. And then I will do that. Play. So is everybody seeing my screen?
Yes, we can see your screen.
Okay. Well, it's a pleasure to attend this and as a speaker this webinar. And as promised, I will be talking about planktonic gastropods. Some of the examples you can see here already. Some of you may recognize pteropods, but there are also another group of gastropods here, which I will introduce later. So yes, Christy was correct. My main affiliation is at Naturalis Biodiversity Center, where I have a research group called Plankton Diversity and Evolution. And I just want to highlight here our wonderful institute. It's a new museum. It's a public museum, as well as it's housing the national collection. So we have more than 40 million objects, from minerals to all kinds of biodiversity there. But we also have about 200 researchers, and I do invite you to contact me if you are interested in doing writing maybe a postdoc grant, or if you want to come on a visiting grant, because we do have grants for visitors who want to use our facilities. And this is in Leiden, which is a very beautiful small city, quite close to Amsterdam. So hereby you are invited. I always like to start by zooming out and taking a look here at our wonderful planet. And I would like to point out, of course, that it is a blue planet, and maybe we should call it water instead of Earth, because it's the largest habitat on our planet. And there's a lot of biodiversity in our oceans. And it is quite surprising that still we know quite little about how diversity in the open ocean arose, how did it evolve, and how does it persist in this seemingly homogeneous space. And this has been the topic of my research already for quite some years, maybe like more than 20 years. But I would like to give away already one of the take-home messages for this talk, is that marine zooplankton have high evolutionary potential. And I published this paper with my long-term collaborator, which many of you probably know, Professor Erika Goetze, in 2013. So that's already 10 years ago. And here we basically pose the hypothesis that marine zooplankton may evolve fast. And this is actually incredibly important, especially considering how fast our oceans are changing because of climate change. But working at a biodiversity institute at a museum, and also I've been trained as an evolutionary biologist, I really care about evolutionary questions. And of course, I would like to just start here with this very beautiful image of Darwin, asking these very basic questions that evolutionary biologists ask, and apply it to the open ocean. So these are my research questions more generally. So how many species are there? How did species evolve? What determines species distributions? And also I'd like to pose the question then, are speciation processes different in the open ocean from land? So most what we know about evolution is based on terrestrial model or organisms, or at least terrestrial species or freshwater habitats. And an important question, since our oceans are changing so rapidly, is how will planktonic species respond to ongoing climate change? And I suppose you all know this background picture of the warming stripes, showing that our Earth is warming because of the CO2. And the anthropogenic carbon release rate is unprecedented since many million years, as we know. So what happens to all that anthropogenic carbon? I think most of you know, but I would like to just show this very basic graph. In red, the increase of carbon dioxide in the atmosphere, and in green, the increased partial pressure of carbon dioxide in the oceans, which leads then to a declining pH, which is a process that we have called ocean acidification. And as you can see, actually, maybe ocean acidification is not really the correct term here, because the oceans don't get acidic, but they get less basic. So their buffering capacity reduces. And this is a problem mainly for calcifying organisms, but also other organisms are affected. And this is the picture is a bit simplified, but it shows that calcifiers have a harder time making their shells. And this is a problem for corals, for instance, calcifying algae like coccolithophores, but also calcifying zooplankton like foraminifers, and the gastropods that I will focus on in this talk. And that's because I've worked on this research question for the past years. I had a major grant, 5 years, to focus on this with a team of people. I didn't do all this by myself. And our main million-dollar question was basically, can planktonic gastropods adapt to ocean acidification? So first, some images of these beautiful organisms. So these are pteropods. They are called pteropods because their foot has evolved into wings, and they fly through the water like butterflies through the sky. And you can see here some shelled pteropods, decoiled pteropods, which are about 1 millimeter in size, and some long spiny ones, which are like they are like half a centimeter. And the one that's swimming around in the dish, that's a sea angel. It's a shellless pteropod that predates on the shell pteropods. So why are they so interesting? Well, to study, they are abundant. They make these shells of aragonite, and that's why they've been termed the canaries in a coal mine, or the most vulnerable, and showing the negative impacts of ocean acidification first. Because aragonite is a highly soluble polymorph of calcium carbonate. And I started to study them actually because they have, as the only extant zooplankton group, they have a fossil record. Of course, planktonic foraminifers have a fossil record too, but they are not like animals like metazoans, like these gastropods. Right. Now I'm thinking, yes, okay. So we have, in the pteropods, we have shelled pteropods, which we call sea butterflies. And you can see here, some are, they have coiled shells, and others have more symmetrical or long and spiny shells. And their sizes range between like 0.5 millimeter to up to 1 centimeter. And then there's the naked pteropods, which we call sea angels, and they are the predators of mostly shelled pteropods. And the sea butterflies, they feed with a mucus web, which can be quite large, like much larger than themselves. And they basically catch particles in these mucus webs with which they feed. The other group of gastropods, holoplanktonic gastropods, that doesn't get a lot of attention in ocean acidification research, are the heteropods or sea elephants. And you can see two examples here. On the right, you can see a shelled heteropod, and on the left there is an unshelled one. And they can be very big, the unshelled heteropods. Maybe you've seen them also in the waters around Asian countries, where they can be up to even 20 centimeters in length. And they are also active predators of, for instance, shelled pteropods and other zooplankton. I did also want to show you this movie that we took on the ocean cruise on the Atlantic Ocean in 2017, which is actually the most beautiful snail I've ever seen. A very big purple sea elephant. The species is Oxygyrus inflatus, and you can see it's cleaning its shell here. Sometimes you can see its radula, it's a rasping tooth coming out of his snout, and he's cleaning his shell. He's looking around. I think it looks like an intelligent animal that's also looking back at us. So can planktonic gastropods adapt to ocean acidification? And I have done this with a great team of people, PhD students and master students and postdocs, visitors. And I'm going to show some data from them. So here I would like to highlight a few people. And I will indicate on the slides who contributed to which data. And I shouldn't forget also Debbie Wall-Palmer, who is unfortunately not in one of these pictures. So one of the things I want to show first is the work that was part of the PhD research of Lisette Mekkes. And she went on a research cruise in the California current ecosystem. And she zigzagged up and down through the upwelling waters that happen there. And these deep waters that come up near the coast are colder and lower in pH. And so she was sampling offshore and onshore stations and examining theropod shells from the species Limacina hilacina using light microscopy, micro-CT scanning, and SEM to quantify calcification and dissolution. Right, and what she found was that offshore, she found that shells were thicker where the waters were less acidic. And she did not find much evidence for shell dissolution. So shell thickness declined by 37% from offshore to onshore. And because we didn't find that the shells were dissolving from the outside, we concluded that theropods make thinner shells in the upwelling region of the California current ecosystem. Of course, this actually leads to a lot of other questions like, would this be a problem? Is it a problem that they have thinner shells? Are they more vulnerable to predators or disease? And also, how long can they continue making thinner shells while these waters are becoming more and more, while these waters are acidifying? What we didn't look at is the genetics. So we don't know whether they were locally adapted genetically or whether there was some physiological adaptation or acclimation. So that would be an interesting future avenue of research. So another approach I took was to zoom out a lot more and look at it from a macro evolutionary perspective, and then ask what has been the evolutionary history of theropods, and were they affected by previous calcification crises in the geological record. And so we know that there have been two major calcification crises in the past. One was about 65 million years ago, the Cretaceous asteroid impact, and one is 56 million years ago, the Paleocene-Eocene thermal maximum. And we know that most theropod fossils originate from after these calcification crises. So that led also to the hypothesis that they are quite a recent group. However, we built a really thorough phylogeny based on more than 2,600 genes. And we combined this with fossil evidence to obtain a time-calibrated evolutionary framework. Well, this tree doesn't look so nice, but this has been published. But what we found, and these yellow horizontal bars, they are the error bars on the time divergence estimate. So you can see there's quite some uncertainty, but we can also say for certain that some of these groups are much older than these previous calcification crises. So for instance, this is the group of the sea butterflies, the shelled theropods, and this is the group of the sea angels, the unshelled theropods, and they turn out to be clearly sister groups that diverged about 140 million years ago, so way before those calcification crises. So this means that they are probably more resilient than we thought on evolutionary timescales. However, this doesn't tell us much about how they will survive the current changes, because what's happening right now with CO2 in the atmosphere is going at least 10 times faster than this group has seen in their evolutionary past. So that's why we also took a more experimental approach. So on one of those Atlantic expeditions, so we've joined the AMT program that's led by the UK, so the Atlantic Meridional Transit program, that goes yearly from the UK down to the South Atlantic. And in 2017, I joined with my PhD student Lisette Mekkes, and we did some experiments. So we did PCO2 treatments, replicating past, present, and future ocean carbonate chemistry levels. And we did short-term onboard incubations to look at calcification responses and also looking at differential gene expression. Yes, so I should say that these, of course, are not completely realistic experiments because, well, you know, those planktonic snails, they don't really like to be in those containers. So when you keep them for longer, they start to become sick and die. So that's why we kept it short. And so in this case, they are not such great like experimental animals, but this could give an insight in how they are coping with these different ocean carbonate conditions, and we can make a relative comparison. So I'm going quite quickly here, just showing a few of the results. Oh, I'm thinking now to hide these. Yeah, I think that's better. So you can see here what we did with those shells. On the left, you can see calcine staining. So it binds to calcium carbonate. And then you can see how much of the shell was grown during those 3 days of the experiment. But since in the museum, we also have very nice micro CT scanners, we could also scan the same shells. And these shells, by the way, are from a heteropod, Atlanta arienseni. And yeah, so these experiments were led by Debbie Wall-Palmer, which is the second publication that's listed here. So using micro-CT scanning, you can quantify the amount of calcified material really accurately, and also you can partition it in the old shell and the newly grown shell. So you can really quantify the amount of calcium carbonate that was precipitated. So overall, what we found was that calcification is negatively impacted with decreasing pH, but that when you provide a lot of food, this had a positive effect on calcification. So one of the things that we saw is that when we were feeding them a lot, they would calcify more than the old shell. So they would make a thicker shell during the experiments, even also in the future ocean conditions. We also found that juveniles were especially sensitive to ocean acidification. And importantly, we found species-specific responses. So not every species responds in the same way. Some species were more sensitive than others. And also it depends probably where you are in the ocean. So I suspect that also the response may differ between different populations. So coming back to these questions, it becomes really important. Oops. It becomes really important if we want to address the question whether planktonic gastropods can adapt to a changing ocean, we need to consider evolutionary processes. So we need to know about species boundaries and to what extent populations are mixing and how populations respond to selective pressure. So I came across this video also on Twitter, so maybe some of you have seen this. I thought it was amazing, showing these incredible swarms of shelled pteropods in the Red Sea. And these are quite a big species, Cavolinia uncinata, so that's like a 1 centimeter shell here. And I just wanted to show, this is quite unique, but these organisms can occur in huge, huge numbers. And I just wanted to highlight here this panmixia paradigm that was around for a long time, although I think many people, including myself, have shown that it's not true for plankton. But the idea was that you have really big populations, high fecundity, high dispersal potential, and relatively few species, and therefore you have slow rates of evolution. But we do know now that we need to revise that panmixia paradigm already since that review paper that I highlighted earlier from 2013, looking at population genetic studies of all kinds of zooplankton, we find in general that a lot of cryptic species are discovered. We find high genetic variability. We find spatial genetic structure. And we pose this hypothesis that selection may be very important. So therefore, I don't think we should view the plankton as some homogeneous, very much mixing entity, but actually what we found, and other people as well, is that many of the populations are adapted to the local oceanic conditions. And I'm going to show some examples of that, and we'll see how much time I have left. But this is a genus that I focused on with my team because they are very abundant. And you can see these are the Limacina pteropods. They are all coiling, but they are left coiling. So that's how you can recognize pteropods very easily. And these are the 5 nominal species. In fact, there are many additional subspecies or actual species within these morpho species. So we have these 3 first species that have warm water circumglobal distribution patterns. And Limacina bullimoides is the most abundant of them. And then we have these 2 nominal cold water species with bipolar or antitropical distribution patterns, so in the northern hemisphere and in the southern hemisphere. So I'll start with the first species. That's Limacina bullimoides, which was the focus of the PhD thesis by Le Chin Chu. Maybe some of you know her. She's originally from Singapore, and now she's doing a postdoc in Sheffield with Roger Butlin, working on Littorina gastropods evolution as well. But for her PhD, Limacina bullimoides was a focal species. And what we know is that they have a life cycle of about 1 year. They are sexual hermaphrodites, and they do diel vertical migration, probably to a depth of 200 meters. And she took a genome-wide approach to look at the genetic structuring of the populations across the world. And we based this on a draft genome, a transcriptome. And so in this way, we could design a capture based on 2,900 probes. And that has been published. If you're interested in this approach, we did this in collaboration with Galice Horo in North University. And we currently actually have a PhD student from the Philippines. Maybe he's even here, I don't know. He's also working with Galice Horo in North University and collaborating with me on the evolution of pteropods. But so this was published this year. I wanted to highlight this study, which I think is very nice. We had several sampling locations in the subtropical, tropical waters of the Atlantic Ocean, the Indian Ocean, and the Pacific Ocean in the North and the South Pacific. And so Lichin screened 161 individuals using this newly designed capture and obtained more than 100,000 SNP markers. So these are single nucleotide polymorphisms across the genome. And they actually show a really clear result that there are 3 distinct clusters. So we have an Atlantic lineage, we have an Indo-Pacific lineage, which occurs in the North Pacific and the Indian Ocean. And we have a Pacific lineage that occurs in the North Pacific and the South Pacific Ocean. So these lineages, we have colored them blue, green, and orange. And you can see that there is absolutely no mixing here. So there are 3 lineages, and they have very specific distribution patterns across the world's oceans. And maybe you noticed already, 2 of those lineages, the green and the orange lineage, are sympatric, so occurring together in the North Pacific Ocean. And they've been distinct since approximately 1 million years. And we looked into these samples more to find out, can we see any morphological differences between them? And in fact, we don't really. We looked at shell shape differences and they are not different. But what we did notice is that the Pacific lineage individuals all had these wing spots. So we know that these are actually spots on the flesh and not on the shell. But we don't know really what they are for. So it could be that they are not cryptic, but they can be distinguished based on these spots on their wing feet. So yeah, please have a look at this paper if you are more interested in more details. And I'm also happy to answer any questions you have maybe now. But for now, I think it could be fun to show also some unpublished data, because maybe you noticed that in the Atlantic Ocean, you have quite a lot of variability there. And so we zoomed in more and screened additional individuals from 14 sites in the Atlantic Ocean and screened them across 97,000 SNPs. And we are still working on this paper, but interestingly, when we zoom into the Atlantic Ocean, we also find these 3 highly distinct clusters, and we don't find evidence of genetic mixing either. So does this mean that there is also 3 species in the Atlantic Ocean? By the way, I forgot to say that we did conclude that those 3 worldwide lineages are definitely distinct biological species that do not interbreed. So now zooming into the Atlantic lineage, the question is, do they really not interbreed? So how come they are not mixing? So one of the things we did is we collaborated with physical oceanographic modelers to look at, if we release particles from these sampling stations, would they have the opportunity in their lifetime to meet each other? So we ran these for 1, 2, and 10 years, and we ran them for different ocean depths. But I'm just going to show you one of the animations here at 100 meters depth. And you can see there is actually some physical retention between those. The barriers are also indicated here in this map. But after about 1 year or so, you can see that they start to be able to mix. So from this, we tentatively conclude that dispersal barriers are permeable. I'm not sure, how am I doing with time? Sure.
It's okay, you may just go ahead.
Okay,
Please.
I'll just finish this story and then yes. Okay. Here it is shown actually the results for about this is after 2 years, and you can see there's actually quite a big difference if you do these physical simulations at 0, 100, or 200 meters. And but basically you can see that these colors actually do get mixed. So they do meet each other in nature. That's basically the conclusion from this. So what keeps these populations apart then? We did also look at abundance in another paper along this transect, and you can also see here, these are chlorophyll images from space. So in blue, those are the oligotrophic gyres. So it seems like the red and the blue populations are more adapted to these oligotrophic conditions, and then the yellow are more adapted to the more mesotrophic conditions in the equatorial upwelling region. And we can also see that the abundance resembles that. So in these barrier regions, they are less abundant. So are they really not mixing was basically the question. And for that, we developed a SNP panel, 30 SNPs, just to be able to screen more individuals. And this work was done by Peijnenburg. And he did an amazing job looking through all my samples and finding more than 700 additional specimens and screened them at these SNPs. And so here in pie charts are shown the samples where we found a mixture of the 2 species or the two colors that we cannot distinguish morphologically. And you can see there are stations where we find both or 2 or 3 colors. And so basically we conclude there is virtually no genetic mixing. We only found one hybrid. I don't know if you can see it, but it's in, if you see my mouse, it's here, it's indicated in green. So we found one hybrid based on these 21 SNPs. But this means basically that they meet in nature, but they don't interbreed, so they are probably biological species. So coming back to that question that I needed to answer, of course, with my grant, can planktonic gastropods adapt to ocean acidification? So I would say probably yes, as a group, but there will be winners and losers, and we definitely see negative effects of ocean acidification. So it is still a question of how they will cope with the ongoing rapid change of our oceans. And I think especially there are some species that are especially vulnerable. For instance, in the polar regions where the changes are also incredibly fast. So as a take home, I would say there is more diversity than meets the eye. Correct species identification is important because we know different species may have different responses to ocean change, and especially also for pteropods because they are used as bioindicators. However, we still actually know a little about the basic ecology of pteropods, and this is important if we want to better understand their responses to ocean change. And for that, I think we have to focus on eco-evolutionary processes to predict better how plankton will respond to global change. And then, of course, I would like to thank all the people who contributed to this work. My funding agency, the Dutch scientific organization, NWO, and the plankton team at Naturalis, as well as my colleagues at the University of Amsterdam. And thank you for your attention and questions. I'm going to stop.
All right. Thank you very much, Dr. Katja. That was a very interesting talk. I think most of the comments I received were they are saying that the pictures are really nice. Yes.
That's
and
very
then
nice.
yes, so I think everybody will be inspired to take photos of their samples now. All right, so we have some questions here. So you may ask your questions, by the way, if you're okay to speak in the microphone, guys. Okay, so we have one question here. He's in one slide, in one of your slides, low pH did not result to reduction in calcification, but the shell seemed thinner or less dense. Can this be related to your other study where calcification increased when food availability was high?
Oh, wait a second. Now I lost you. Wait a sec. So can you repeat the question? I was looking for my presentation and then I got lost. So yes.
Okay. So sorry, can you hear me clearly?
Yes, I can.
Yes. Yeah. Okay. So a question here. So in one of your slides, low pH did not result to reduction in calcification, but the shell seemed thinner or less dense. Can this be related to your other study where calcification increased when food availability was high?
I'm not sure which slide he refers to, but I was giving an overview there about from different studies.
Yes.
And for instance, I'm just thinking what he's or she is meaning exactly. So definitely food availability is very important. So I think it can counteract the negative effects of ocean acidification. But in our experiments, we always fed our pteropods. So they were quite happy with that. So overall, they were calcifying quite a lot. But then we were comparing relative amounts of calcification. So the shells were never, I think in one study, though, we published in Frontiers in Marine Science, we did find that the density indeed was less in present and future conditions compared to the past. but it was always a relative comparison between the treatments. Yeah, so if you compare the old shell with the new shell, then I think we did that. We didn't do that for all the studies, but then you could see that overall the new shell was thicker. But maybe we should really look into the actual structure of the shell, right? Like we didn't really do that. The microstructure, how are the crystals built? And it could be that they are less dense.
Okay, thank you. Yeah, so I guess you were
I hope I
presenting two
answered the
different
question.
papers then. Yeah.
Yeah.
Okay, so for the first time, I guess we have a question from a live audience here, from the live audience.
Great.
So we have Dalvin Carlo Chan Lamawan. Please, you may turn on your microphone, please. Yes, Dalvin, please unmute. Dalvin, you may unmute your microphone, please. Oh, maybe we are having a problem with the microphone. Okay, maybe we have another question, I guess. Who was that? So anyway, we will go back to you later, Dalvin. Okay, so we have other questions here. Would you consider thinning of theropod shell an adaptation mechanism to ocean acidification? Or is it an influence of, or the other way around?
Well, that's a very good question. So is it an adaptation? It could be, because you don't want to make a thicker shell if that's really energetically costly, or if you don't need a thicker shell. But the problem is that the ocean acidification is continuing. So I don't think that they can go on making ever thinner shells, because at some point there will not be a shell left, and they do have a shell for a reason.
Yeah, otherwise they're going to be naked. Naked.
Yeah. Well, maybe that's going to be the ultimate. Although I would like to point out the naked theropods, they also have shells as juveniles, so they are also still equally vulnerable, probably. Yeah.
Okay, so we have a lot of undergraduate students in the audience, and one question is, what do the swarms of Cavolinia mean, or what does that indicate? Because in most samples we have a lot of copepods, but your video showed a lot of Cavolinia.
Yes, well, this is quite special, I think. I've never seen it like that, but I know that people have also observed it in like other tropical regions. I think it is like, it depends on the environmental conditions, and then you can have a swarm, especially those bigger theropods, they can swarm. In general, I think theropods are quite abundant, but you also have to fish with quite, well, we fish with a bongo net, which with quite a large mesh size, so that you can have some speed. So if you do very gentle vertical tows, I think you find less theropods.
Oh, they will escape. Okay.
Yeah.
So
yeah,
should
they
be
can escape.
quick.
Yeah, they can swim a little, you know, with their wings.
Wings. Yeah.
Yeah.
All right. Thank you. So we have another question. Can you culture theropods and do mesocosm experiments with induced CO2 aquatic acidification? And if I may add, there is a 6-year-old girl in the audience, and she's asking, how long do theropods live after collection in the lab?
Wow, thank you so much for these questions. Well, we don't really know how long theropods live in the open ocean. Maybe it is up to 1 year, but it could also be less, especially in the warmer water. It could also be more. I mean, it could be 2 or 3 years. I have to admit that when we catch them, they usually don't live so long, because they are already damaged by the net. So they, in the experiment, they would live up to 1 week, 2 weeks. But then yeah, they also need a lot of water. So mesocosm experiments would be the answer, I think. And in Woods Hole in the United States, they have managed for one species to keep them in the lab. So I'm not sure if they can actually reproduce, but they can grow them up from the egg until the adult. But it will require like a lot of resources, like fresh seawater, and yeah, you have to spend a lot of time and energy, I think, to try and keep them alive in the lab. But I can also say that in some aquaria, like I've been to Japan, where they have an aquarium. In Osaka, they have the sea angels on display, and they can keep them, I think, for a year, but I think after that they also die.
Okay. Yeah, so for sure this will make the 6-year-old girl cry.
Oh yes, I'm sorry.
Another
Well, the
question
good news is
yeah.
the good news is that there is a lot of plankton, you know. So I do we take quite a small sample of the plankton to study it. And also we preserve it in the museum. All our samples are accessible in the museum so that people can look at it later.
All right, thank you. So another question on the transcriptomic study on Atlantis, did you find differentially expressed genes related to calcification?
Yes, we did. Yes, of course. So we don't really know exactly what genes are responsible to make the shell, but we have genes that are good candidates for making the shell, and we do see in general some upregulation of these genes in the future conditions. But you have to look at the paper because it's not so straightforward. The response is not so easily explained. A lot of genes are up or down regulated, and indeed we do find indications that the calcification process is affected by the treatments.
So another question, I think this is from an undergraduate student. From Chu et al. 2023 paper, it is surprising that there's no mixing in population. Do you think it is mainly driven by differences in nutrient and thermal regimes?
Well, good question. So indeed, there's not much mixing happening between the different species, but because they occur in the same waters, I think that's not really driven by the environmental factors, probably. I think they are real species, but we can't easily see the difference. But they can see the difference, right? Like they recognize each other by maybe chemosensory. They recognize each other as distinct species. So I think they might have a different ecology. So they might have a different depth habitat, because we didn't do any depth stratified sampling. We just made one big plankton hole. Or they might have different trophic ecology. They might eat different things, and we don't know these things.
All right. So in your global study or observations, you saw that the land barriers were really effective. So, but in your zoomed in experiments, you saw that there might be some little mixing, but there was only a hybrid in one of the SNP stations, right? So it seems that the even the water boundary or barrier is still effective in separating the different species.
Well, yes and no. I think the currents, they show that they can actually mix between the water masses. And you don't need a lot of mixing for the genetics to be mixed. So I think they actually occur in the same samples, but then they still don't interbreed.
Okay.
So actually, I think that is good evidence to say that they are real species.
Separate species, right?
Separate species, indeed.
So maybe we have 2 or 3 more questions. Sorry, have you tried using RAD sequencing to explore population structure?
No, I haven't. So we chose to use the capture approach, which is also another approach to get like reduced representation sequencing. So we have also SNPs, but with rat, those SNPs are anonymous. So you don't, I mean, we didn't, if you have a really big genome size that we have, you would need a lot of sequencing to get enough coverage for that amount of variability. So that's why we chose the capture approach, which is a bit more costly, and it takes time to design the markers. But when you have it, you also have more information about the protein coding genes. So we are also planning to look more into that.
Excellent, thank you. Maybe this question is really important for a lot of us here in the Philippines. How many species of theropods are there, and is there any available literature for the identification down to species level, morphologically?
Wow, yeah. I have to say that the information is quite scattered in different papers. And so how many species are there? Well, about 60, 70 species, something like that. Yeah, so there is not really, I mean, I think I like the book from Lally and Gilmer, 1989, Pelagic Gastropods. I think it's a really nice book to know more about them. There are some good overviews. So if this person sends me an email, I can send some overviews.
All right, so please send Dr. Katja an email, guys. All right,
Yeah.
so is Lally and Gilmer the same author for the biological oceanography book, Lally and Parsons? Is it the same book? Okay,
Could be.
never mind.
I'm not sure. Yeah, it could be.
Yeah,
I don't know.
okay, that's interesting. Okay,
Yeah.
thank you very much. Do we still have more questions? Oh yeah, we have a question from an anonymous attendee. Why would the upwelling lead to lesser calcification in theropoda? Upwelling would bring nutrients and it will enhance primary productivity, thus increasing food.
Yeah, that's true. So in this upwelling region, the deep waters come to the surface, which indeed are colder and they bring nutrients, but they also have lower aragonite saturation states, and so they have lower availability of carbonate minerals, and that's why we find thinner shells there.
Okay, and then maybe the last question. What is or are the reasons why onshore region have lesser calcification? Is it because of runoffs and high mixing in the onshore region?
Yeah, so the problem with doing this kind of research in the natural environment, you have natural gradients, is that all these different ocean parameters are correlated. So we can't say for sure what is the driving parameter for thinner shells. So it is in the upwelling region we find thinner shells, and the waters there are colder, and they are less buffered. So they have lower alkalinity, lower carbonate, lower aragonite saturation state. So basically it can be, and lower pH. So basically it could be either the carbonate system, so lower pH, or it is lower temperature. But it's not food. There was not a strong correlation with chlorophyll.
Okay, thank you. Do you have final words for the audience before we move on with the program?
No, well, thank you very much again, and thank you for all your nice questions. And also, really, I mean, I'm serious about that you can contact me if you have additional questions, or if you are interested to maybe come and visit the Netherlands and do some research in my team. I would be happy to support you. So that's what I wanted to highlight.
Okay, thank you very much. We have a lot of students who are working on plankton now. So I guess it is a very good opportunity to join your lab. So guys, hear this announcement.
Okay.
All right, so thank you very much. Now we shall award the certificate to our speaker. This is a certificate of appreciation awarded to Dr. Katja Peijnenburg in recognition of her outstanding contribution as keynote speaker of presentation titled Evolution of Planktonic Gastropods in an Acidifying Ocean, the third lecture series on Philippine Planktology on the 17th of July 2023, signed by the director of the Institute of Marine Fisheries and Oceanology, Dr. De Leon, and me. And then Dr. Louis Lopez, the administrator of the Philippine Planktology page. Thank you very much again for joining us today. It's very early in your where you are right now. Maybe 9 o'clock or 8 a.m.
Yes.
Yes. Thank you so much for spending your time with us. And I believe that a lot of people are very grateful for hearing your talk. Thank you for the inspiration.
Okay,
All
my
right. So
pleasure.
yes, thank you. Before we end, I would like to plug the different degree programs that is offered in the Institute of Marine Fisheries and Oceanology. I think we're still accepting applications. So please log on to our website. And also please follow these Facebook pages where we have different announcements of webinars and other activities for undergraduate, master students, and other graduate students in the Philippines. Thank you so much again. So please watch out for our third and the last but not the least lecture from Dr. Leo Cajo Bershal from Bermuda. And this will be on August 29. And this is, I think, at 8 a.m. because he is in a different country. And so we have to adjust to his schedule. Thank you very much. I hope everybody has seen the evaluation sheet. All right, so it's there in the chat box. You may now get the link. Thank you very much, Dr. Katja. Maybe we can have a photo, please. I think this is only for the panelists. Can I ask Elise to do the? Sorry, wait, here. Okay, stop sharing. Yes. All right. So may I ask Elise to do the screen capture, please? Who else is here? Oh, by the way, we have around 110 participants, both from Zoom and also on our Facebook Live.
Okay, great.
Yes, thank you very much. And we have participants from abroad, also from Thailand and Taiwan. So quite interesting.
Cool.
All right, so are we ready, Elise?
Yes, ma'am.
Okay. Okay, thank you very much, Dr. Katja. We hope to see you in one of the conferences for plankton.
Have a nice day.
Have a nice day. Louis, thank you.
Thank you.
Everyone, staff of Info, and everyone else who worked hard
Thank
for
you.
the lecture series. Direct, thank you. Bye-bye.
Bye.